Hello, and welcome to Talks with Interesting People. Today I'm joined by Dr. Richard T. Marconi, a physician microbiologist researcher and professor at VCU, Virginia Commonwealth University, here in Richmond, Virginia. Dr. Marconi has spent more than 3 decades studying Lyme disease and other tick-borne infections. His work has helped shape our understanding of Borrelia burgdorferi and other tick-borne diseases, including anaplasmosis, ehrlichiosis, and babesiosis. His research has led to the development of widely used canine Lyme vaccines and is now driving innovative approaches towards next-generation Borrelia and tick-borne disease diagnostics, as well as human vaccines. Dr. Marconi is at the forefront of translating complex infectious disease science into real-world solutions, making him uniquely positioned to help us unpack both the biology and the controversy surrounding Lyme disease today. So thank you so much for joining us today, Dr. Marconi.
Well, thank you for having me. Pleasure to be here.
Awesome. So to start with, I want to hear kind of about your background. What got you into this area of research? What was your major area of study in school? And you're a
Sure.
physician researcher, so how did you end up transferring into primarily research?
Well, just to be clear, I'm a PhD researcher. So,
Oh, are you PhD MD?
PhD scientist.
Oh my gosh, I've gotten this backwards all along. My apologies. PhD microbiologist. Thank you.
Yes. Well, my interest in Lyme really began obviously several decades ago. I grew up in Brooklyn, New York, and then moved out to New Jersey when we were, around 1970, and really got to enjoy nature and in the great outdoors. And interestingly, while New Jersey is an area now that is highly endemic for Lyme, as a child who spent every day outside, I do not recall a single time ever seeing a tick, or of course hearing about Lyme disease. But was always interested in the outdoors and nature. And when I went to college at William Patterson College in New Jersey and studied biology, after leaving there, I went to the University of Montana to study microbiology, and I completed my PhD there. And after leaving the University of Montana, I came back to the East Coast where I continued to study bacterial metabolism at the Roche Institute for Molecular Biology. And while I was at the Roche Institute during my postdoctoral training, that was in the late 1980s, there was an interesting article that appeared on the cover, I believe it was Newsweek magazine, that talked about a mysterious illness that was spreading across 43 states. And that mysterious illness turned out to be Lyme disease. And I got very interested in that and decided to do a second postdoctoral training stint focused on Lyme disease and other tick-borne diseases. And to do that, I decided to go to the National Institutes of Health Rocky Mountain Laboratories, which brought me back to beautiful Montana. But the exciting thing about going back to Rocky Mountain Laboratories, or going out to that particular lab, was that the real pioneers in the early days of Lyme disease were working there, including Willy Burgdorfer, for whom the bacterium is named. So I had the honor of interacting with Dr. Burgdorfer while I was there, and many of the other key, early players who were involved in the discovery and characterization of Lyme disease. So that's what got me interested in it, and I've continued my focus on Lyme and other tick-borne diseases after joining Virginia Commonwealth University in 1994.
That's wonderful. I didn't know you had to study with Dr. Bergdorfer himself, so you've been in this since the beginning.
I came in a little bit after the beginning,
A
but
little
fortunately, I got to work with the people who were there at the very beginning. So,
Yeah, that's
yeah,
awesome. If you wouldn't mind, just broadly, we'll go into this far more deeply. Describe your current focuses of in research.
sure. So my lab is very interested in a lot of different bacterial diseases and tick-borne pathogens. So while a large focus has certainly been Lyme disease, as evidenced by our publications in the development of a Lyme disease vaccine, we're also interested in other tick-borne diseases, which include ones that you may not have heard much about, but which are very, very important and certainly on the increase. And that includes diseases like anaplasmosis and ehrlichiosis. Both of those are bacterial infections transmitted by ticks. And then a parasitic infection called babesiosis, which is also transmitted, in fact, by the same tick that transmits Lyme disease. And in addition to the work that we do on tick-borne diseases, we're very interested in a pathogen called Leptospira. Leptospira is the most common zoonotic infection worldwide, and we're putting a lot of emphasis there to develop both vaccines and diagnostics as well.
That's awesome. I'm going to jump straight into it. Can you tell us about Borrelia, why it's a unique bacteria and, about the different types? And are you guys specifically focused on Borrelia burgdorferi? And yeah, so I'm interested in about why it is a unique bacteria and all the different forms and which ones you guys focus on.
Well, yeah, that's a great question, and I could speak to that for days because it really is a remarkably fascinating bacteria. It's very different from most other bacteria. It belongs to this unique group of bacteria that we call spirochetes. So other examples of spirochetal bacteria would include things like the causative agent of leptospirosis, the causative agent of syphilis, and spirochetes are also very important and play a very significant role in periodontal disease in both humans and animals. So it's a really fascinating group of bacteria. And the reason we call them spirochetes is because they have this really unique structure. They basically resemble a corkscrew, for lack of a better term. And that structure is actually really important because it allows these bacteria, when they transmit from a tick or enter into a mammal, to very efficiently drill through tissues and disseminate throughout the body. So you can think of a corkscrew, if you turn a corkscrew in midair, it goes nowhere, but if you put it up against a matrix like a cork, in turn it will drill. And the spirochetes do that. So that's a really interesting and unique feature of them, but they're also really interesting at the genetic level. And this is a really important point because it's something that was not appreciated for a long time in the early days of studying Lyme disease. And the point I'm making is that these bacteria, for example, the Lyme disease spirochetes, vary significantly in nature. There's a lot of different variants of the Lyme spirochetes. In fact, when I started working on Lyme disease, there was one species that was known to cause Lyme disease in humans, and that was called Borrelia burgdorferi, again, named after Willy Burgdorfer. My work at Rocky Mountain Laboratories focused on analyzing the genetic diversity of these bacteria. And we and others had proposed that there are actually multiple species of bacteria that can cause Lyme disease. And that's now well recognized. So in the United States, the primary species is Borrelia burgdorferi. Then there's also another one called Borrelia mayonnaise. In Europe, we have a couple of additional species. So it's a really diverse group of bacteria. And recognizing that was a critical step because in order to make effective vaccines and diagnostics, you have to account for that natural diversity that occurs in these bacteria in nature to make effective products to either prevent or treat or diagnose the infection.
Yeah, so now I have a million other questions. So, I think I'll start with, are the Borrelia bacteria changing as rapidly as other bacteria in response to antibiotics? So like do we have different subtypes of Burgdorferi and is it Maiemoti?
Yeah, so there are two
Yeah.
different questions really. So the Lyme disease spirochetes, Borrelia burgdorferi being the species, the example you're using, is not really changing a lot in nature. So it's a pretty stable organism, although there's a lot of diversity in these bacteria in nature. With regard to, the use of antibiotics to treat Lyme disease, et cetera, there have been, to my knowledge, no reported cases of antibiotic resistance that develops in the Lyme spirochetes as a result of the use of antibiotics. So unlike many other bacteria where we have to be very concerned about that, that does not seem to be a significant factor with the Lyme disease spirochetes.
That's one piece of good news.
Yeah.
I hadn't heard that talked about much. So kind of two different questions then. So that might lead into one of the others. Like, so why is Borrelia so controversial? Why is it so controversial in diagnostics? And then we'll get to other treatments. Why is it so controversial? Why is it so hard to treat? But I think that has to do with they call it sort of the ghost. It's hard to find in the body. So which leads a lot to the controversy. So yeah, the broader question around the controversy of understanding Lyme in conventional medicine, and specifically why it's so tricky to diagnose.
Well, focusing initially on the question of why it's difficult to diagnose. The infection can at times be fairly easy to diagnose if certain clinical presentation is observed. So for example, the development of that bullseye rash, as we call it, or erythema migrans. Obviously, when that type of rash develops, it's a really clear indication of the possibility of Lyme disease. So in many ways, people who develop that type of a rash are somewhat fortunate because it helps direct the clinician toward what the causative agent of the infection is. Unfortunately, or I guess you'd say unfortunately, not everyone will develop the rash. So as a result of that, they don't seek out treatment as quickly as someone who might have developed a rash. Obviously, if you have an expanding bullseye rash develop on your body, you're likely to seek treatment for that and get diagnosed earlier. But when that doesn't occur, it leads to a later diagnosis, and that makes treatment a bit more difficult. Now, diagnosis, there are tests available, as you're aware, for Lyme disease. But the problem, again, is that the tests that are currently being used don't really account for all of that incredible diversity that I talked about that exists in the Lyme spirochetes. So an individual that's infected with one particular strain of the Lyme spirochetes might be easier to diagnose than an individual infected with a different strain, just because of the differences in the bacteria themselves.
So can you tell me then more about like the two-tiered testing and your thoughts on how that is used? And what is the primary version of Borrelia that it tests for?
So the two-tier testing that's currently in place was really designed to test primarily for Borrelia burgdorferi, which again is the dominant species associated with disease in North America. The test is very useful, and it certainly plays a very important role, but it can miss positives. So you can get false negatives. You can potentially get false positives. The other inherent limitation of two-tier testing is evident in the name of the test itself, two-tier. We really want to have a test that is a direct, simple, one-stage test to diagnose Lyme disease. And that's where two-tier testing is sort of lagging behind a little bit because it requires multiple tests to come up with a diagnosis. Whereas for most bacterial infections, that's not the case. We have pretty straightforward testing for them.
Why is it the testing need to be two-tiered for Lyme? And if you could explain the science behind the ELISA versus the Western blot and why it's done that way for Borrelia, because I can't quote any other infections that's used for it.
Yeah, so the initial test in the two-tier testing is the ELISA test, as you referred to. And there's some variations on that test as well, but generally it's an ELISA-based test. And basically it will screen for antibodies to many different components of the Lyme spirochetes. So when you get infected, we will make antibodies to the different proteins produced by the bacteria, right? In an ELISA test, you're taking the bacteria itself in most formulations, and you're putting it into what's called an ELISA test, and you're using that to screen for antibodies. Now, it's helpful, that ELISA is helpful, but the problem is it's not highly specific. So generally, if you get a positive ELISA test, they will then advance to an immunoblot test, which is considered to be more specific than the ELISA. So that's why the two tiers are in place. Again, there's certainly room for improvement there. And do we really need two-tier testing down the road? I think that's an open question that we should think about. I think we can improve testing to the point where we don't have to do two-tier, but we can develop a single test that will validate a diagnosis or confirm a diagnosis.
That will lead into, I want to hear about what you're doing for diagnostics. But if you don't mind explaining a little bit more about the two-tiered testing, as far as like what stages of Lyme infection would they maybe best be able to help identify?
So one of the things that you often hear about testing for Lyme disease is that the tests are not reliable during early stage infection. That's one of the key areas that you'll hear about most. And there is some validity to that because whenever we get infected with any infectious agent, it takes our body some degree of time to mount an immune response. Right? So typically, you'll start to get antibodies that develop as a result of an infection, about 7 or 8 days into the infection. But with the current tests, you don't get detectable levels of infection reliably until you're several weeks out. So as a result of that, serological testing for Lyme has been criticized a lot. But in reality, I think serological testing is highly valuable. The problem is that we're not testing for antibodies to the right proteins, because there are certain proteins made by the bacteria that are made at very high levels during the earliest stages of infection and do in fact elicit enough antibody that can be detected, 10 days and 7 to 10 days into an infection. Another important thing to note about diagnostics is that most people don't come to a clinic or seek treatment until they're well beyond that time point anyway. They're typically coming in after symptoms develop, and that takes a couple of weeks generally, with the exception of that bullseye rash that I referred to, which can develop pretty quickly. So the criticism, that the serological tests aren't working earlier enough, well, there's some caveats to that, and I think that's important to keep in mind.
Awesome. So is that a good segue into your research? And if I'm going to quote it correctly, the OspC protein?
Well, so yeah, that is a good segue. So let me tell you a little bit about OspC. OspC is a
Tell me
protein.
everything.
Okay. So it's important to understand that the Lyme spirochetes live in very different environments during their cycle. So they live either in a tick or in a mammal. They cannot live freely in nature. They have to live in association with a host. So you're not going to find them in water. You're not going to find them in soil, or you're not going to find them on a surface, etc. Now, as you can imagine, the environment inside of a tick is a very different environment than the environment inside of a mammal. And the analogy that I like to use is to say that when you go outside on the coldest day in the winter versus the hottest day in the summer, you're going to change your clothing to adapt to the environment. The Lyme spirochetes have to do the same thing. So when they're living in a tick, they wear a different set of clothes than they do when they're living in a mammal, right? So there's two proteins that have been really focused on primarily for vaccine development efforts, and those proteins belong to this category of proteins that we call outer surface proteins. And we abbreviate that, we call them OSP proteins, for outer surface protein. So I talked about how the Lyme spirochetes have to change their coat in these different environments that they live in. When a Lyme spirochete is living inside of a tick, it coats itself with outer surface protein A, or OspA. And it has to have OspA in order to survive in a tick. However, once the tick starts feeding, in other words, taking in a blood meal, the spirochetes no longer need the OspA protein, and they stop making it. And they switch to producing a protein called outer surface protein C, or OspC. So once that change occurs, where the spirochete switches from making OspA to OsC, the bacteria is then able to migrate out of the tick and enter into the tissue of a mammal and establish an infection. And it turns out that this outer surface protein C is the 8th most abundant protein that the Lyme spirochetes make when they get into the tissue of a mammal. So that was really a very important piece of information for us to utilize when we sought to develop a vaccine for Lyme disease. Unfortunately, OspC is very difficult to use as a diagnostic marker or diagnostic antigen because it is a very highly variable protein. So we know worldwide that there's about 35 different variants of this OspC protein. So to have, while you would think OspC would be useful as a diagnostic because it's made very early during infection, it mounts a good antibody response. The problem is that the antibody response that develops to OspC is very, very specific for the version of OspC carried by the infecting strain. So it's hard to make a diagnostic that can pick up antibodies to all the different variants of OspC that exist.
You mentioned it's the 8th most prolific, so those first 7 are not good candidates either?
They are not. So when I talk about the abundance of a protein that's being made, I'm talking about the proteins made by the bacteria in general. So remember, bacteria make proteins, many proteins stay within the cell. They're not exposed on the surface.
Okay.
So those proteins are not really useful in diagnostics because they're not seen by the immune system,
Okay.
at least not during early infection.
Okay. So you say I guess that would segue into so many other questions. I'm trying to pick one, excuse me. So then the development of vaccines out of OPSI, tell me about your research in that. And I know for a while there was a human Lyme vaccine, and so I would love to know about it in veterinary science, veterinary use, and then tell me what happened with the human vaccine.
Sure. So I'll start back by talking about the one and only human vaccine that entered the market for Lyme disease. And this was a vaccine that was called the Lymerix vaccine. It entered the market in 1998. It was pulled from the market in 2002. So it had a very short tenure, in terms of its usage. Now, part of the reason it was pulled from the market was that there were perceived concerns about adverse events being elicited by vaccination. Now, interestingly, that was looked at rigorously and with, outstanding teams of investigators looked at that and it was determined that there were no adverse events elicited by the vaccine itself. But nonetheless, once that information had sort of become public domain, people became concerned about using the vaccine and demand for the vaccine dropped. So the vaccine was actually removed voluntarily by its manufacturer. It was not removed because of any proven health concern. That vaccine consisted of the OspA protein that I mentioned just a few minutes ago. So one of the medical issues with the vaccine, with that particular vaccine, was that the vaccination schedule required that you got 4 doses of the vaccine within the first 14 months that you are using it. So that's a very rigorous vaccination schedule. And generally, as you know, with vaccine hesitancy and concerns that people have about vaccines, unfortunately, that was a tough sell. And in addition, even after 4 doses of the vaccine, the protective efficacy of the vaccine was about 72%. Now, 72% is not a bad number for a vaccine, but it's not a great number for a vaccine that you have to get 4 doses of. So I think one of the interesting things about the Lymerix vaccine, again, which was an OspA vaccine, is why didn't it work better? Or maybe the better question is, how did it work at all? Because if you recall, I mentioned that the OspA protein is only made by the bacteria when they're living inside of a tick. So
Yeah.
most vaccines work because we vaccinate, we develop antibodies, we develop immune memory cells that can respond to a pathogen, a disease-causing agent, when it enters the body. And then our immune system is able to tackle it once it enters the body. Well, OspA is not made when the bacteria enter the body. So how did the vaccine work? The way that it worked is that if you were sufficiently vaccinated such that you had high levels of antibodies to the OspA protein circulating in your blood, when a tick fed on a vaccinated individual, the blood goes into the tick and the antibodies are in the blood. And if there's enough antibodies, it can kill the bacteria or prevent them from transmitting from the tick into the vaccinated individual. The problem is that with all vaccines, after you are initially vaccinated, your antibody levels will drop over time. That's a natural, immunological phenomenon. So if your antibody levels drop below a certain level, there's not enough antibodies to the OspA protein in your blood to prevent all of the spirochetes, to prevent all the spirochetes from moving from the tick into the mammal. So any spirochete that's able to make the passage from the tick into the mammal, because it's now not producing OspA anymore, even though that individual is vaccinated and has antibodies to OspA in its blood, the antibodies can't touch the bacteria because they don't have any OspA on the surface. And that's also the approach that is being taken now with a vaccine that has just completed phase 3 clinical trials. It's a vaccine being developed in a partnership between Pfizer and a company called Valneva. And they have a modified form of an OspA-based vaccine. After completing phase 3 clinical trials, the recommended dosing for that vaccine is, again, a 4-dose schedule. So again, in today's climate, one of the questions I have, how well will that be accepted by the public at large? Will it be covered by insurance? will people, consistently get the boosters that they will need? So there are a lot of questions yet to be answered about that. That vaccine is not yet on the market, but it's anticipated that the companies involved will probably move forward for FDA approval, I think sometime in 2027.
Why is it approved and working in animals?
So at the same time that the LymeRix vaccine was developed for human use, it basically also got applied for use in veterinary medicine, specifically in dogs. Um, and it's certainly, as I said, it's certainly safe. It's a very safe vaccine. It's a vaccine that consists of just that single protein, that OspA protein. So it's a nice, clean vaccine, and it is effective if you're able to sufficiently boost an animal and maintain the levels of antibody at a high enough level. I mean, when we set out to make a new vaccine for use in humans, we actually started by working on vaccines for humans. But because of the failure of LymeRix, the market was simply not there. Big Pharma had very little interest in moving on another human vaccine at the time that we started working in this area. We started then working on a vaccine for use in veterinary medicine that would be an improvement over the current vaccines that were available. And there are several vaccines on the market for use in veterinary medicine. There is that OspA vaccine that's called Recombitek, but there are also a couple other vaccines that are called bacterium vaccines. And a bacterium vaccine is a very crude type of vaccine. Basically, we could make one here in my lab by taking a frozen stock of the Lyme spirochetes out of the freezer, growing it up in the laboratory. We would then collect the bacteria from that liquid culture. We would kill them because we don't want live bacteria in a vaccine. We would homogenize the bacteria, emulsify them, filter them, because you don't want clumps in a vaccine. And then that would be your vaccine. So a bacterium vaccine is a vaccine that consists of everything that the bacteria make when they're growing in the laboratory. And we know that the Lyme spirochetes make about 2,000 proteins when they grow in the laboratory. Yet only a couple of those have ever been shown to play a role in eliciting protective immunity. So there's these two types of vaccines. And again, the OspA vaccine, which is called a recombinant vaccine or a subunit vaccine, and then the bacteriums. So when we set out to make a new vaccine, we clearly didn't want to make a bacterium. We wanted to make a subunit vaccine that's nice and clean. And we recognize that OspA is a useful component to have in the vaccine. Because if you have OspA in the vaccine, you can elicit antibodies that will at the very least inhibit some of the bacteria or prevent some of the bacteria from moving from a tick into a mammal, right? But we really needed something else in the vaccine that could kill the bacteria when they actually get into the vaccinated mammal. And for that purpose, we utilized OspC. Again, as I mentioned earlier, it's a very highly produced protein by the bacteria when they get into a mammal. So as a result, there's lots of OspC on the surface of the bacteria, and that's good because it means there's a lot of target for your antibodies that you've induced by vaccination to target on the surface of the bacteria. The problem with using OspC in a vaccine, I also alluded to this earlier, is that it's a highly variable protein. So with 35 different variants of OspC out there, how do you come up with a version of OspC that will elicit antibodies that can target all of those different strains, making all of the different OspC proteins that are in nature? And the solution we came up with is we decided that nature wasn't going to hand us the answer. So we had to create our own answer to how to make a broadly protective version of OspC. So to make a very long story short, I'll tell you that what we did is we took the OspC protein, we identified the parts of it that actually stimulate the antibody production during an infection. So we found these small pieces of OspC that are the immunologically important parts of the protein. And we call those parts of a protein epitopes. So epitopes are the immune stimulating part of a protein. So what we did is we just created a new protein in which we took the epitope parts from many different OSCs and assembled them together into a new protein. So the second protein in our vaccine is not really an OspC protein. Instead, it's a protein that is based on OspC, that is the best parts of many different ossies assembled together. And we call that type of a protein a chimerotope, which stands for chimeric epitope protein. And a chimeric is when you join things together. And what are we joining together? We're joining together the epitopes from many different ossies. And that's what led to the vaccine that's now on the market that is called Vanguard CR Lyme. That's produced by Zoetis. And we were using that same approach that we used to develop CR Lyme to advance a human vaccine, as well as to develop vaccines for other really important tick-borne diseases that most people may not have heard of, that include things like anaplasmosis, ehrlichiosis, and babesiosis.
That's awesome. Yeah, I've definitely heard of Vanguard for my dog. Let me see. I think that I had so I was the word you used is bacterium. I would say that that's basically like OG vaccines. That's kind of how they first did it, isn't it?
Yeah,
Just grind it up
yeah,
sterilize it
essentially,
and just
yeah.
inject it. Okay, yeah.
That's true, but I want to be fair to the manufacturers that they go a little bit beyond that. So they do, in fact, try and
Okay.
clean them up a bit.
Yeah.
But nonetheless, I think the key thing to recognize about quote unquote bacterium vaccines is that they have so much stuff in them. And
Yeah.
there's a lot of proteins in there, and all of those proteins are going to trigger an antibody response. But a lot of the antibodies that get triggered by some of those proteins, they don't play a role in protective immunity. So
Yeah.
when you use a bacterium, you're getting this misdirection of the immune response. The immune response starts to focus on proteins that are not really important in providing protection. So in a subunit vaccine, you're putting in proteins that you know elicit protective antibodies. So it's a much more focused type of vaccine that elicits a much more focused type of antibody response.
Yeah, it sounds like the Bactrim ones could, we all know what it's like when we got vaccines and we felt kind of flu-like after. I figure you'd feel pretty icky after the Bactrim ones.
I don't think that I wouldn't want to say that because
Okay.
to my knowledge, there's no evidence now, at least we're talking now again about canine vaccines, that the Bactrim vaccines are eliciting any adverse events.
Oh, and
So
I don't mean adverse vaccine events. I just mean like normal flu-like after a vaccine.
in dogs, we certainly notice that they can be a little bit sluggish after
Yeah.
vaccination, as any
Yeah.
animal would be. But
Yeah. Yeah.
they don't articulate it as well as people do.
No, they'll be okay.
Yeah.
Um, I want to get into the tick-borne co-infections, certainly, but if you
Sure.
don't mind, first, just while we're still on Borrelia, why all of the controversy between conventional medicine, often who would only recognize a short-term recent acute Lyme infection, and there's no such thing as chronic Lyme or post-infectious Lyme, versus, the kind of the other end of the spectrum where kind of everything is chronic Lyme. And so what's being misunderstood about Lyme's behavior? And I guess another question is why is Lyme so evasive to the immune system? So I think which is I think part of it.
Sure. Um, First, let me talk about the issue of whether Lyme disease is a chronic infection or not, because I think this is really important. And I think the term chronic often gets misused. So the Lyme spirochetes, when they infect an animal, they establish a chronic infection. They can persist in an animal, or it could be a human, a dog, a horse, wildlife, for a very long period of time. I'm talking about years. So they're actively in that animal, they're replicating, they can be recovered from the animal. So there is such a thing as chronic infection, right? But I'm talking about chronic infection in the absence of treatment. So let's focus on this chronic infection issue for a minute. If you think about for lack of a better term, the life cycle of a Lyme spirochete, as I said earlier, can only exist.
When you say chronic infection, you may not be necessarily referring to chronic illness. You're just saying that you have the spirochete in your body. It may or may not result in actual illness for the
That's
animal.
right.
Okay,
That's
sorry,
right.
go on. So you
All
said
right. So when you are chronically infected, as I said, the spirochetes can persist for a long period of time, years. And this has been shown in humans, it's been shown in dogs, it's been shown in wildlife. But it only makes sense that the Lyme spirochetes would cause a chronic infection because in nature, they can only live in two different environments, a tick or a mammal. So they have to constantly cycle between the two because in humans, there's no human to human transmission of Lyme disease. There's no animal to animal transmission. The only way that a mammal can get infected with the Lyme spirochetes is if they're bitten by an infected tick. Okay. Now, in ticks, ticks go through a very specific developmental cycle, starting with eggs which hatch and the larvae emerge, and then the larvae develop into what are called nymphs, and then the nymphs develop into the adult stage ticks. Now, there is no transoviral transmission from an adult female tick of the Lyme spirochetes through the egg. So in other words, all larvae, when they emerge from an egg, they are infection-free. So the only way a tick can become infected is to feed on an infected animal. So you keep those 2 points in mind. Ticks can only become infected when they feed on an infected animal, and there's no animal-to-animal transmission of Lyme disease.
I
Right?
don't want to interrupt, but is there maternal child transmission? That's been one thing that's talked about in the Lyme community.
Yeah. So, that's a good question, and it's something that definitely still needs investigation. It's been looked at in some animals. For example, we looked at it in dogs. We saw no evidence there of intrauterine transmission. But again, placental structure is very different in a dog versus a human. So there does need to be more work done on that. I would say at this point in time, there is not strong evidence that there's transmission from the mom to the fetus, but I don't think that that's completely conclusive. So.
Thank you. Sorry. So back to the nymphs, or excuse me, the larvae are infection-free until feeding on an infected mammal.
Yeah. So the question is why has evolution turned out this way? Why is it that the Lyme spirochetes can cause such a long-standing infection in an animal? Well, I think the answer actually makes just perfect sense, and that is that if you think like a Lyme spirochete for a minute, if you infect an animal, what you want to do is stay in that animal for as long as you possibly can. Because for your population to continue in nature, you need to have a tick come along, feed on that infected animal, pick up the spirochetes, and then go feed on another animal and pass the spirochetes to another mammal to maintain the cycle of Lyme spirochetes between ticks and mammals in nature. And we call that cycle the enzootic cycle. So the spirochetes need to maintain a chronic infection to ensure their survival. But also related to that is when a Lyme spirochete gets into a mammal, it doesn't want to destroy its home, so to speak. So Lyme disease tends to be a slowly developing disease. It can be very, very debilitating, but it generally takes time. It's not necessarily a really acute infection with immediate, clinical manifestations that develop. And again, that only makes sense because, as I said, they have to cycle in nature. They don't want to get into an animal, destroy that animal, and then not be able to be picked up by a tick and passed on to another animal.
Awesome. I mean, so that brings up a question, and I'm just my own questions that I'm making up as I hear you talk. We don't test for like PCR bacteria in blood very often. Because I was taught that you can't find Borrelia that way, and yet the main route of transmission is a simple tick sucking your blood. What's going on there with my inability to make those two things
Sure.
make sense?
Well, so the Lyme spirochetes actually survive quite well in tissue. And they can really achieve higher densities in tissue and in the organs. But you're correct, you don't see a lot of them in the blood. But when a tick feeds, it's not just drawing up blood, it's drawing up, fluids from the tissue as well. So that's how the tick becomes infected. Now, you can find spirochetes in the blood of an animal shortly after it is initially infected, but they're at very low levels. So PCR of blood is in fact quite difficult and not particularly reliable for detecting the Lyme disease spirochetes.
Is there other tissues that not very clinically useful that could be better used for diagnostics for Borrelia spirochetes?
Well, the problem is that utilizing other tissues can be quite invasive, right?
Yeah.
So for example, in veterinary medicine or in the laboratory, when we're studying infections in animals like mice, for example, we will collect tissue biopsies, small, punch biopsies of skin tissue. And the reason we do that is because the spirochetes are living so well in the tissue. If you're able to collect a small skin punch biopsy and put it into the culture media that we use to grow Lyme spirochetes, you can readily grow the spirochetes from that tissue biopsy. But that's generally not an approach that's going to be widely used in human
Yeah.
medicine. And in addition, while you can grow the spirochetes from something like a tissue biopsy, they grow very slowly. So it can literally take weeks to get a positive culture, and that's not very useful, in human medicine.
Okay. Okay. So let me see. So then in conventional medicine, no, people go to their doctor, they run an ELISA, it's negative, even if they, have positive symptoms or positive exposure, and they're kind of sent on the way, maybe given a few, sometimes one-time dose of doxy as much as 28 weeks. What is that missing? And then kind of a huge question would be, of course, we can think of infections living in the body chronically. I mean, there's many viruses that live in our body forever. What do you think? I'm asking too many questions at once, but one would be kind of more on why is diagnostics so difficult and confusing, and the next one being what would allow some people to become very ill from Lyme, maybe from an infection from kind of long ago, and other people seem to be able to live with it just in their
Sure.
body without being ill.
Well, some of the questions that you're asking, we don't have answers to yet, but I will offer some thoughts on them
Yeah,
nonetheless.
that'd be
So,
great.
um, all individuals are different. Our immune responses are different. No two humans are alike. Similarly, no two ticks are alike. As much as we like to think of them as being, this one entity, there's a lot of genetic variation, biological variation in ticks. And there's also this very biological and genetic variation in the Lyme spirochetes themselves. So some strains of the Lyme spirochetes are more prone to causing invasive, more severe infections than other strains are. So that can influence the course of an infection and how serious the clinical manifestations may be. The other issue is that it's really important to remember that even after someone is treated for Lyme, you can be potentially treated successfully for Lyme disease, but you can still have, in a significant number of cases, long-term effects as a result of the infection. So you can have sequelae from the infection. And these things can persist for long periods of time. They do tend to generally go away, not in all cases. But I think that's a key distinction to make, because we know from so many diseases that the infection can be very taxing on the body, but you can treat it, but you can still have long-lasting damage that resulted from the infection. And you're going to be dealing with those symptoms potentially for a fairly long time. And, the inclination would be to say, well, I have Lyme disease. I have these symptoms, I had a tick bite. Well, and you may very well have had Lyme disease, but do you have an active infection at that later stage? Maybe not. It may just be the consequences of the infection.
Yeah, that's honestly when I get into discussions with different people in the tick-borne disease world, I'm kind of on this, we can't, of my understanding, definitively prove that the infection isn't still present in the body or other infections, but I'm kind of big on the theory that there are certain environmental triggers that lead to immune dysregulation long term. It doesn't have to mean that there's an active infection going on still.
Yeah,
And I know in the Lyme world, they believe that, and pardon if this offends anyone, but like they believe that if you're sick, that means there's a tick, there's an infection that needs to be killed with antibiotics. So yeah.
I agree with you. So I agree with what you're saying. As I said, you can be very effectively treated, but it doesn't mean that there wasn't damage that you'll have to deal with. And of course, the most difficult thing for any patient is not having a definitive diagnosis, no matter
Yeah.
what the illness is. Living with the unknown and living without answers is really tough on people, and understandably so. And sometimes those patients who are suffering from long-term sequelae of an infection, that's unfortunately what they're dealing with. There's no clear-cut diagnosis to pin to describe what they're going through. And
Yeah,
then,
that
oh, go ahead, please.
oh no, go ahead.
I think it's also really important to think about the possibility that an individual who's bitten by a tick may be infected with other agents that are not responsive to doxycycline.
Well,
So
let's segue.
yeah.
Anaplasma, Ehrlichia, Babesia. Yeah.
So fortunately, some of these other pathogens are quite responsive to doxycycline and other common methods that are used to treat Lyme, antibiotic-based methods. So anaplasmosis, ehrlichiosis, your doctor may think you have Lyme disease because you were bitten by a tick. They give you doxycycline. You get better. But maybe it wasn't actually Lyme. It could have been anaplasma. It could have been Ehrlichia. It could have been one of these others. But Babesia, or babesiosis, as the disease is called, that's a parasitic infection that's transmitted by the same ticks that transmit Lyme disease. And the organism Babesia, what it does is it gets into the body and it infects red blood cells. And obviously, having parasites living in red blood cells is not a good outcome for an individual. And unfortunately, Babesia is not responsive to treatment with doxycycline. So, you could go into a doctor's office, for example, have a Lyme disease rash. You could have Lyme disease, but you could also have been co-infected with one of these other agents. Maybe you will respond well to the treatment for Lyme disease, but not to the presence of one of these other pathogens, whether it be Babesia or some of these very serious viruses that are starting to show up in ticks. So, that could also account for some of the patients that have these long-term effects. They're really ill. Maybe it's not Lyme, but it could certainly have been transmitted by the tick that fed on them some time ago.
And are you developing diagnostics around these other co-infections, the three that we've mentioned?
We are. So our focus right now is both on vaccine and diagnostic assay development for a human Lyme disease vaccine. We've already achieved the veterinary vaccine. We are working now on vaccines for Ehrlichia, Anaplasma, and Babesia, and new diagnostics for all of them. And in all cases, we're using that chimerism approach that I mentioned earlier because it has many inherent advantages over fairly traditional ways of developing vaccines and diagnostics.
And so where are you in the pipeline for the development of that? And is it more veterinary or animal, probably that you'll think it'll come out for?
Well, so,
Veterinary.
obviously, when you develop a new product, you have to encounter the business side of it as well, right? So you have to get, a company that's going to pick up and run with it. One of the reasons that things come out quicker in the veterinary world is because we can get approval a bit quicker. It's not less rigorous, but we can get it quicker. We also have the ability to directly test how well products work, like vaccines, in animals that we cannot do in humans. So, for example, if we develop a new vaccine for any of these tick-borne diseases, we can vaccinate an animal, and then we can come back and actually challenge the animal, feed ticks on that animal that are carrying the disease, and determine whether the animals are protected or not. It's very direct. In humans, that's not an option.
Yeah.
So it's a very different approach. It takes longer to do it in humans. It's significantly more expensive. The liabilities are significantly greater. So that's really why veterinary medicine moves faster, just because of the ability to sort of do direct testing of how well something works.
Yeah. This is a little bit off the immediate scope, but there's some use. Have you tried to translate your work at all with, I know it's a virus, but like with COVID and long COVID, especially with some of the possibilities that COVID vaccines can help people get over long COVID, the idea of kind of creating immune tolerance post-infection. So have you applied any of your research ideas to COVID? And then also, would you think that a Lyme vaccine might be able to have the similar effect in a post-Lyme infection person to help them create more tolerance to the ongoing immune response?
So we have not done any work on COVID. And the reason why is my laboratory is largely focused on bacterial pathogens, with the exception of Babesia, which is a parasite. So the world of studying viruses is actually very different than the research world studying bacteria. So we are focused very exclusively on the bacteria slash parasite side of things and have not delved into COVID directly. So.
You think, and so the second part of that question is, do you think that a Lyme vaccine could possibly work to create more tolerance in a post-Lyme infectious person who's still on dealing with symptoms that seem to have started after that infection?
So if your question is, can vaccination have a therapeutic effect? The answer to that is none of the current vaccines that are used in veterinary medicine. Remember, there is no vaccine right now for humans, so we can only talk about veterinary medicine. There's no evidence that any of the current Lyme disease vaccines, including CR Lyme, there's no evidence that they are therapeutic. And actually wouldn't expect certainly CR Lyme to be therapeutic because of the following reason. CR Lyme consists of two proteins, OspA and that OspC chimeratome, right? OspA, since it's not made during an infection, can't serve as a target for a therapeutic response because there's none of it on the surface. OspC, even though it's one of the most abundant proteins made by the bacteria during early infection, it tends to be not produced during later stages of infection. The Lyme spirochetes are adapting to their environment. They're changing the proteins that they put on their surface. So OspC is a very good protein for eliciting an antibiotic response to prevent an infection, but it's not necessarily useful for a therapeutic impact.
Okay, awesome. What do you think, research-wise, what's the best way to diagnose Lyme? And then what do you think is the best clinical way? I mean, so I have ELISA, I have Western blots, there's this different specialty DNA connections, IGeneX, there's all kinds of specialty tick-borne testing labs, and I have a hard time trusting the research and science around it. Like, why would this test be positive and this is negative? So from a pure research perspective, what do you think is the best diagnostics? And then if you have an opinion on what might be the best clinical.
Yeah, so that's actually a difficult question, but let me try and provide some thoughts on it. Certainly, one of the most important aspects of getting a handle on whether someone has Lyme disease or not is assessing the patient's history. So first of all, have they been in an area where Lyme disease is endemic? Even if it was for just a day, have they traveled to an area where Lyme is endemic? Do they live in an area where Lyme is endemic? So for example, you're very unlikely to get Lyme disease if you're living in Arizona. There's not a lot of Lyme in Arizona. If you are in Virginia, the Northeast, it's an endemic area. So right away, you have to start from the position that it is quite possible this patient could have gotten Lyme disease, right? Then you look at the clinical presentation. Well, early stage disease doesn't give you a lot of true markers that are really indicative of a Lyme infection, with the potential exception of the bullseye rash, right? So if they have that bullseye rash, that's very helpful. Often that's enough for a clinician to make a diagnosis and move forward with treatment. Unfortunately, as I said earlier, and it seems odd to say unfortunately, the rash does not develop in all patients, and it doesn't always look like a bullseye. So it can often look like some other type of arthropod bite, a spider bite, etc. But you look at that information, you look at whether the person has a history of, or do they recall being bitten by a tick? That's important to sort of assess. And then, I think I'm not going to specifically say that one test is better than the other. I think there are weaknesses in all of the tests. So I think adhering to the fairly standard approach right now of two-tier testing is unfortunately probably the best thing we have. There are a lot of companies out there, I guess I would call them fringe companies, trying to make money off of Lyme diagnostics. One needs to be very careful about those companies because their tests have not been fully validated. So that's not a direct answer to what is the best way to diagnose Lyme disease, but I do think serological testing is very, very useful, coupled with clinical history, and we absolutely need to improve testing. Yeah, there's no question about that. So we don't have the ideal test just yet.
maybe in an attempt to dispel or bring some light into some of the popular things in the some of the Lyme communities is, practitioners will diagnose Lyme off of a positive IgG 23, 53, 93, 41. And my understanding, because my background is more in physiology rather than clinically, is that just shows that you have potentially been exposed to the organism. And then also, IgM, you'll see IgM become positive in people who are fine, or it's not anywhere near an infection, any possible infection. So number one, your opinion on diagnosing Lyme based on IgG proteins being positive, and then the idea of how and why the IgM is positive long after the case, after infection.
Yeah, I'll start with the IgG. So based upon, the early years of studying Lyme disease and the proteins that it makes and how mammals as a whole respond to infection with the Lyme spirochetes, it was determined that there was a set of about 10 proteins that could be considered as good markers to detect antibodies to an infection with the Lyme spirochetes. And with IgG, the current classification is that if you are positive for 5 of those 10, it is considered a positive diagnosis for Lyme.
And that's just CDC criteria, yeah.
CDC.
Yeah.
Now, that set of 10 proteins, you referred to them by number. That's just indicating what we call the molecular weight of each of those proteins. Some of those proteins have other names. But the problem that I have with that set of 10 proteins that are being used to help diagnose Lyme disease via IgG responses is that I think there are actually better proteins that could be used. So I don't think the set of 10 that's currently at play is the optimal set of proteins. Let me give you an example. You referred to P23. P23 is actually just another name for OSPC.
Because that's one I was told is pretty highly specific for Lyme.
Well, so the problem is, and we have published this, and so have many others. If you take one particular variant of OSPC, we call these different variants of OSPC types. And they've been given letter designations because we have to be able to talk about them. So we'll refer to OSPC type A, OSPC type B, OSPC type C, et cetera. There's 35 different types, so you run out of letters, and then there's some other designations. But the diagnostic tests only have one version or one type of OSPC on the immunoblot. And we know, for example, that if you get infected with a Lyme spirochete strain that is producing OSPC type A, for example, you will make a very effective antibody response against OSPC type A. But the antibodies won't bind to the other OSPC types. So if the OSPC that's on that immunoblot is OSPC type B, for example, and you get infected with a strain making a different OSPC type, you could get a negative result for OSPC, even though you are in fact antibody positive for OSPC. And
we need like 35 23s to match all the different variations of the OSPC.
I'm sorry, repeat that, please.
So we need, you'd mentioned that there's about 35 varieties of OSPC, and
Right.
so we need like 23 different OSPC, or 20, we need 35 different 23s to match the different OSPC variations.
So yeah, in reality, you probably wouldn't need all 35 because some of them,
Yeah.
some OSPCs will elicit an antibody response that can recognize a few of the other versions of OSPC, but you would need to have some critical mass, just the right number, the right, types of OSPC that in order to pick up an infection with any strain. And that's one of the key things that we had to do when we designed CR Lyme. We had to make an OSPC that would be able to elicit antibodies that could kill any of the different strains making different versions of OSPC. So we need to bring that same type of approach into diagnostics. And we're doing that. So for example, we have developed a diagnostic antigen for Lyme. It is still in validation and in testing in our lab. But this particular diagnostic antigen or protein actually has pieces from 4 totally different proteins joined together. And the reason that we did this is if you take parts of several proteins and join them together and you use that to detect antibodies, you're not dependent upon detecting antibodies to just one protein. Your chimeric can now detect antibodies that might have been elicited to multiple proteins, right? So we think that's advantageous. We think it increases the sensitivity of testing and also the accuracy of testing. And ultimately, I think it's very possible, although we're not there yet, that through the development of chimeratopes for diagnostics, we can get rid of two-tier testing, which I think would be a big step forward. As I said, two-tier testing is what we have now, but it doesn't mean we can't get a lot better.
Yeah, absolutely. I mean, science and medicine, the thing when people always are like, okay, we think we know everything. And remember what we didn't know 20, 50, 100 years ago. And you think we're not going to know equally more, if not more, in another 50 or 100 years.
And it
Yeah.
doesn't even have to go that long. I can think back to, things and to concepts or to conclusions we made about data 10 years ago.
Yeah.
But now we've learned things that say, no, actually, that wasn't the right interpretation.
It
And
speeds up. Yeah.
that's the important part of science is recognizing that, you don't know everything and you have to take in the information as you go and modify your train of thought.
So I like talking with researchers because they'll never tell you this is what's going on. They'll say, this is what we have found thus far. We can say
Exactly.
this might prove this, but we need to follow up. Yeah.
Yeah, exactly.
I know this is more kind of an immunology question, but about the IgM being positive,
Okay.
long after I'll have someone sitting in front of me perfectly fine, are doing a lot better, and they'll still have one or two positive IgM bands. I'm not about to run them through Lyme treatment.
Sure. So IgM is tricky. I mean, as you're probably aware, when antibodies develop, the first form of antibody that develops in the body as a result of exposure to a foreign agent is IgM. But IgM is not very specific. So, you get IgM produced early, but we can't use it alone to diagnose Lyme disease, although there is a recommendation that if 3 of 5 bands are reactive with IgM. I'm not fully confident in using IgM to make a diagnosis. I'll be quite honest about that. Now, something you indicated that in some patients you'll see that the IgM can persist, right? And you'll have it still reacting with some bands on that immunoblot strip. Well, IgM really doesn't persist that long, but it's important to remember that something else could trigger an IgM response. And because of the lack of specificity of IgM, that IgM could be binding with those proteins on the Lyme immunoblot. So there's a lot of possibilities there, but there is a reason why IgG is considered a better and more reliable antibody to use for diagnosis than IgM.
Okay, awesome. So like cross-reactivity, does that have to mean cross-reactivity with like an infection that we need to pay attention to? If the person isn't feeling well, what if they feel okay, but you'll see people come from long-term Lyme treatment and their IgM's still positive and they're better. So is there some other infection we need to think about? And of course, having tested for all the immunoglobulins, making sure they're just not like total IgM high. But there are some, I'm aware of a couple, 2 or 3 other infections I could think of off the top of my head that I've heard have cross-reactivity with Lyme.
So there's some concern that there could be cross-reactivity between the IgM that you might detect in a Lyme test with IgM that could be elicited by things like syphilis or periodontal, some of the periodontal bacteria. But there is no clear cut, in my opinion, at this point, no very specific disease or infection that is more likely than any other to elicit antibodies that would yield a false positive with an IgM test.
Okay, so make a clinical diagnosis and don't necessarily treat the immunoblot.
Well, I'm reluctant to say about what you should do about treatment. I mean,
Right. Excuse me.
yeah,
Mhm.
but as a non-clinician, I do know that, in my opinion, is that if a patient has otherwise been healthy and not having problems, and they come in and they are testing positive for Lyme by these various tests, yeah, I think treatment is very important. It's interesting. In veterinary medicine, they screen for tick-borne diseases every year. In endemic areas, most dogs will be tested for Lyme, Anaplasma, Ehrlichia, and then a disease called heartworm. So we screen them. And a dog may be negative for these pathogens year after year, but then if they come in, let's say at the beginning of this year, and they test positive for some of these pathogens, you're absolutely going to treat because you know that they haven't, this is potentially a new infection because they never tested positive before. We don't have that information in human medicine because screening is not part of the strategy for prevention, which is unfortunate. And there's probably a lot of reasons why routine screening is not done that are beyond certainly our control.
Yeah, absolutely. Yeah, I know I give my dog a pill for fleas, ticks,
Flea and tick
heartworm.
control, sure. Yeah.
Yeah, yeah, yeah. So did you want to speak any more about well, I'm not sure if you're able to speak to, what percentage of ticks, I'm sure that's regional, are infected with some form of Borrelia versus the co-infections? Because I think I was taught that co-infections are um, happen less often.
So first of all, what percentage of ticks are infected with the Lyme spirochetes? Definitely a regional question. It
Mhm.
varies quite a bit. So in the Northeast, in the Mid-Atlantic, and the Upper Midwest, um, the percentage of ticks can be very, very high. Upwards of greater than 90% of nymphal stage ticks or above. Remember, the larvae are not infected until they feed, right? So it's
Mhm.
going to be very high in endemic areas. And consistent with that, if you go into regions in the Northeast and you do serosurveys on animals like mice, which are common reservoirs for carrying the Lyme spirochetes, there are studies that have shown that in certain areas, 100% of the mice that are tested are antibody positive for the Lyme spirochetes. They're probably
Mhm.
also actively infected because once they become antibody positive, there's nothing to really clear the infection. You're not treating mice, right? So, um,
Yeah.
and we've looked at these tick-borne diseases in wildlife. We published a study in which we looked at eastern coyotes from the state of Pennsylvania. And you may ask, why would you be interested in eastern coyotes? Well, I'm interested in coyotes because they're interesting animals, but they're also wild canids. They are closely related to dogs. So by studying these tick-borne diseases in wildlife, we can get a lot of information because wildlife has never been exposed to flea and tick control, as you mentioned a moment ago, right? They've never been vaccinated for any of these illnesses. So you can actually see, what happens in these animals when over the natural course of infection, right? Because there's no intervention that's ever gone on. And interestingly, when we screened 300 and some serum samples collected from eastern coyotes in the state of Pennsylvania, 70% were positive for Lyme, strongly positive. So, if you put a number on the antibody response, they were very strongly positive. 72% were positive for anaplasmosis. So, again, you wouldn't find those same numbers if you looked at small mammals or coyotes in Arizona. But in areas where Lyme is endemic and these ticks are endemic, those numbers are going to be quite high.
And I thought anaplasma and Ehrlichia usually come with quite a severe flu-like illness.
They certainly can. They
Yeah,
certainly can.
potentially deadly.
Oh, absolutely. And I don't want to overstate this about Lyme disease, but I want to be clear that there are published reports that were published in highly reputable journals several years back that Lyme disease can contribute in some cases, can cause death in patients as well, particularly when things like Lyme carditis develop. So, inflammation of the heart. It's rare, so I don't want to overstate
Yeah.
that. But all of these tick-borne diseases can in fact be very serious. And even though Lyme is generally a slowly developing disease, that's not always the case. There's no one size fits all. Some people will develop symptoms very, very quickly. And then, of course, there's a set of pathogens that we never talk about that it can really be problematic, and that are the viruses that are carried by ticks, like Powassan virus and Heartland virus. Just 2 or 3 days ago, there was a warning that came out from the Pennsylvania Department of Health about the rather significant increase in the number of emergency room visits as a result of tick-borne diseases. And they specifically, in that report mentioned Powassan is showing up in more and more cases, still low numbers, but Powassan and Heartland virus. And those are potentially deadly infections.
Do you have any idea what the frequency in anywhere near us of those viruses is in ticks?
In our area, I do not. We don't
Okay.
have that kind of data certainly from Virginia, but
Yeah.
in Pennsylvania, where and other northeastern states, the testing is more rigorous
Yeah.
because the tick populations are higher. So you can find pretty good information on those other infections by looking at the Department of Health websites from those particular states. And they'll break it down and tell you, what counties have the highest number of cases, et cetera. But this brings up a good point that I'd like to make. I mentioned earlier that we do yearly screening for some of the most, common tick-borne diseases in dogs. That's done in veterinary medicine. We actually have probably better databases on the risk of tick-borne diseases in dogs than we do in humans. And there's an organization called the Companion Animal Parasite Council. And if you get on their website, you can click on Canada or the United States. You can click on a particular disease. It'll show you the percentage of dogs in a given state that are testing positive for Lyme. You can click on a county and get the numbers for that individual county. So that data is actually really useful, and I think it would be great for human medicine to look more closely at that data, because obviously if dogs are getting Lyme disease, it means we're at risk for Lyme disease in that area. So that Companion Animal Parasite Council data is really useful, and it's updated every month.
Companion animal, what was it?
It's called the Companion Animal Parasite Council. So
Okay.
capc.org.
And it's not just parasites. Or
Uh,
parasite meaning anything that infects.
it doesn't cover everything, but it covers Lyme, Anaplasma, ehrlichiosis, heartworm, and then a few other infections. But
Okay.
it's not fully comprehensive, but it's a very useful source. CAPC gets their information from the primary companies involved in diagnostics in veterinary medicine, which
Yeah.
include IDEXX, Antech, and Zoetis. Those are the three big ones. But I'll make one point that's really important for viewers to know if they go to the CAPC data. So in 2025, there was somewhere around 700,000 positive antibody tests reported for dogs in the United States. CAPC only collects about 30% of the test results or test data that are done. So if you extrapolate that through, the actual number of dogs that are testing positive each year is going to be probably closer to 1.5 million.
Okay.
So those numbers, you have to keep that in mind. And again, what shows up in animals are sentinels for what's going to happen in humans, whether
Yeah.
it's looking at wildlife or looking at our companion animals.
Okay. Yeah. No, that is really interesting. I'll definitely check out our area.
Yeah,
Um, just in respect of your time, I'll have a couple of rapid fire questions if that's
sure.
okay. I mean, so first of all, is there anything we haven't talked about that I haven't asked you about that you want to make sure that we cover that's an important topic you think related to either Lyme and tick diagnostics and treatment in general, or your research specifically?
Well, there's a general comment I would make, and we've touched on it a few times, and just very briefly, I think it's very important to consider when evaluating a patient whether they may have been infected with other pathogens transmitted by ticks. I think that's a critically important point. Lyme is the one that gets the most attention, but we just can't put blinders on. I think that's the only issue with regard to tick-borne diseases. So that's critical.
And would you say the Anaplasma, Ehrlichia, Babesia, would you also say the viruses, like what are maybe your top ones that you would say should be checked? Of course, depending on symptoms and
Yeah. So I think Anaplasma, Ehrlichia, and Babesia are all critically important. In fact, Babesia is so important that in states where the ticks are endemic, the Ixodes scapularis ticks that transmit Lyme disease and Babesia, blood banks test for Babesia. So when you have testing occurring at the blood bank level, you know that it's a disease of concern. So, we need to do better though. We need to have tests that can be used in the doctor's office or be rapidly done in a clinical setting to diagnose these infections like Babesia.
And do you trust kind of typical LabCorp Quest immunoglobulin tests for these or?
As I said, I think they have decent tests. I would not say that I'm 100% confident in every result that comes out of one of those tests. But at the current time, we don't have alternatives. So
Yeah.
even though they have some weaknesses, we can't ignore their strengths. But again,
If,
we need to make better tests.
yeah, if it's positive, would I trust that versus if it's negative, I might not totally trust it? Is that what I might understand?
I think if a patient tests positive with a test that has been evaluated and carefully designed, and that's going to be the case certainly with LabCorp and Quest, et cetera, that you can have a fair degree of confidence in a positive test and make a decision about treatment.
Okay, awesome. What's the most exciting thing coming in Lyme research in the next 5 years, or if you want a different timeline, that's
Well, so I mentioned that there's a vaccine that's being evaluated for use in humans that's just completed phase 3 clinical trials. Um, I have some concerns about that particular vaccine, but I think it's very positive that there is now at least a push towards developing vaccines that can be used in humans. So I think that's important. I think, ultimately we want to have a couple of choices with regard to vaccines, and we'd like to have vaccines that don't require 4 boosts or 4 immunizations to work. I think we'll get there. I think that the new technologies that are available for doing science have allowed us to much more rapidly assess the variability in Lyme disease strains, genome sequencing, et cetera. So I think that we have the information coming into our hands that's going to let us make better diagnostics as well. And I think that's very
that's
exciting.
exciting.
Yeah.
Yeah. And is that also kind of where your research is going, or where do you see your research going in the next, do those things parallel as far as the diagnostics and finding the different genomics and proteins, or
They
do you
do.
have some
So
other areas you see yourself going?
yeah, I mean, as I said, we're working on both diagnostics and vaccines for all of these infections. The reason I think it's so important to work on both simultaneously is because for example, working on Lyme disease, um, the data that we get can go into one of 2 pots. The data can suggest that a given protein or a given piece of information will be useful for making a vaccine. The other information might be useful for making a diagnostic. The important point is that if you work on them simultaneously, you can make diagnostics, for example, that will not yield a false positive when someone gets vaccinated. And that's
Yeah.
really important because
Yeah.
when vaccination does start to pick up, we're going to have to be able to differentiate between a vaccinated individual and an infected individual.
Yeah, you're thinking way ahead.
It's not that far ahead. I think,
That's
I think I think we'll get there. And I'll just tell you, to me, the ultimate goal of what we do is, at least on the vaccine front, is not necessarily to develop the best vaccine for Lyme disease, but to develop a vaccine for tick-borne diseases. I think
Mhm.
that's really what we want. So, I mean, I want it to be the best vaccine for Lyme, but I want to make a vaccine that will protect against Babesia, Lyme, Ehrlichia, all in one shot, not or
that
1 or 2 shots. And I think that is absolutely feasible with the chimeriscope subunit vaccine approach. We're not there yet, but to me, that is the ultimate goal.
Wow, I had never even thought about that. That would be exciting.
Mhm.
Yeah. Um, One of the last questions, why do you think Lyme is so controversial and misunderstood in diagnostics and treatment?
Well, I think it's controversial because of the weaknesses that currently exist in diagnostics, and also because of the variability that can occur with regard to the clinical presentation. There's and the other part of the controversy is this issue that which we talked about in the beginning, that you clearly can have been infected with Lyme disease spirochetes and been successfully treated. but there still can be consequences of the infection. So the controversy there is, well, do they have Lyme disease or not? Well, we define having Lyme disease as being actively infected. So I think one way we can resolve some of this controversy is not to get too hung up on that and instead understand that you don't, you can be treated well for Lyme, you can respond, but now we have to deal with the other consequences and not blame it on an active infection, but rather as damage from the infection and work on treating that directly.
Oh, awesome. That's perfect. Yeah, I really, really appreciate it.
Sure.
Any other final thoughts that you want to make sure people have take-home messages about your work or Lyme or the co-infections specifically?
Yeah, I would say that it's really important for people to be aware of tick-borne diseases and to encourage everybody involved to support research in this critical area, whether it's Lyme or other infectious diseases. We're at a turning point in this country, and research is under fire, and we can't make people's lives better if we don't have the support that we need to advance research. And that's a critical, critical point.
And what's the best thing people can do to support that? Is it voting? Is it donations? Is it?
It's all of the above.
Yeah.
Certainly, advocating for policies that will promote research and will, sort of not turn your back on what science can offer. That's critically important. Donations are also critically important. I know that my lab personally has benefited by private donations. I'm not soliciting for donations here. We gladly take donations when they're available. But for all of science, it's good to make it an effort that everyone gets involved in any way they can, whether it's just advocating for research or actually trying to support it.
Yeah, science is real. It's a process by which you can, if anyone has questions about that, just talk with researchers. They'll never tell you, unless it truly is an established fact. They'll always kind of say, this is what we know so far. This is connected to this. So, science is the one thing we can hopefully all believe in. So, and I know quite a number of researchers who have been significantly had funding cut and who rely on private donations. So I will solicit for donations then.
Yeah, I will too.
Yeah, yeah,
But
please.
I didn't want to make that the point of my discussion.
I totally understand. I appreciate it. Well, thank you so much for your time. This has been invaluable. As I've mentioned, this podcast is somewhat selfishly for me because I so enjoy getting to talk with experts like you and to educate and to learn and hopefully get some information out so that people can have better informed choices about their life and their healthcare. So I can't thank you enough.
Well, thank you for that, and thank you for what you're doing, because promoting awareness and bringing this to people's attention is critically important. So we're both doing the right thing.
We'll keep trying. Yeah, and I'll follow you. And if you have any updates, let me know. Well, I would love to talk again.
Yeah, please check out my LinkedIn page. I post publications there and other information as it becomes available, and interesting videos for people to look at of ticks
Oh,
and
yeah.
other. Yeah, so
Okay, I will get on LinkedIn and follow you. Thank you so much.
okay. Thank you.
Awesome. Thanks, Dr. Marconi. Take care.